Having me on this in the symposium. I really appreciate it. It's great to be back at EMGS this year, even if virtually. I'm going to be speaking about heritable impacts of general anesthesia, which I consider to be an urgent matter for genetic toxicology and autism research. Mental disability eligible autism in California. If we look at the broader autism spectrum as defined by the DSM, in New Jersey, we're seeing 5% of all boys having autism. And as Dr. Walter showed, it's estimated to be 1 in 54 or even 1 in 40 if you look at some other data in younger children. We've seen this also globally in industrialized countries. The prevalence is now about 3% of children, for example, in Northern Ireland, Japan, and South Korea. But it's quite a puzzle. No somatic environmental factors can account for it, although there's some small contributors, for example, prenatal valproic acid or extreme prematurity. And it's highly heritable, but it's not been shown to be highly genetic. In fact, clinically speaking, only about 10% of autisms can be attributed to a genetic cause. That number may grow somewhat, but it certainly will not account for all the heritability. There's a huge amount of mystery there. So I'm introducing to you a new hypothesis that I think could explain some of this, and it is that general anesthetic gases can, in some cases, induce molecular perturbations in the germline, resulting in dysregulated transcription of genes important for offspring brain development, explaining a subset of the autisms. And I talked about or wrote about this very recently in the journal Environmental Epigenetics with my colleague, LaDonna Ford, and you can get that online if you want to learn more after my talk today. So why this hypothesis? I have to say that this hypothesis is completely left field to the way that mainstream autism research is proceeding now. Mainstream autism research is almost entirely genetics focused, or they're looking at proximal somatic exposures like air pollution or some other kind of prenatal drugs like antidepressants or something like that. But so this is completely from out of left field. So why? And I have to say, it really came from stories I heard from autism families after I questioned them to death. Also, after I started digging into it, discovering general anesthesia's many toxicities, including genomic toxicities. The experimental evidence in rodent models is growing, and I'll talk about that. And finally, it checks out with many puzzling phenomena we are seeing in autism research. So let me start by just talking a little bit about synthetic volatile halogenated anesthetic gases, which is really what, there are all kinds of anesthesia, but I'm focusing on that in this talk. There are many kinds that have been introduced. They started really kind of coming into use in the 1960s, and they've increased in prevalence. I don't have a graph, unfortunately, but I know that they've increased in prevalence markedly since that time, and now they are very prevalent today. Halothane was the first one. Now sevoflurane is the most widely used. These are small lipophilic molecules that passively diffuse through the cells and also in germ cells, and they enter the nucleus. They are essentially a lethal poisonous exposure in the method and in the doses used clinically. They cause global suppression of the nervous system, basically an induced coma, and patients are kept alive artificially through breathing apparatuses and through careful monitoring, for example. They are almost always used in some sort of mixture with other anesthetics, for example, with nitrous oxide or intravenous or neuraxial or local anesthetics, which can enhance the toxicity. And interestingly, higher concentrations need to be used in very young children because of the immaturity of their nervous system. Their GABAergic system works very differently. It's extremely prevalent. We now see about 48 million surgical inpatient procedures a year, and the vast majority of those will use these gases. So why? I mean, listen, I know that family stories aren't science. It's just anecdotes, and it's just stuff that I hear, and it may or may not mean anything. But I want everyone to understand why I'm so interested in this exposure and in this hypothesis. So you kind of have to go into my life a little bit. I'm an autism advocate. I know hundreds of autism families, and I talk to them all the time. And this pattern just could not be, escaped. I couldn't escape seeing this. So I'm going to share with you 4 patterns that I've seen. One was when an autism parent was exposed in utero, i.e. when the mother of that parent, so the grandmother of a child, had to have, for example, emergency surgery for an appendicitis or for some other reason related to the pregnancy or other reasons. And in those cases, there seemed to be an increased risk for multiplex autism in the offspring when the parent was exposed so young. And as a makeshift reality check across all of these patterns, I asked if their unexposed siblings also had kids with autism, and almost to a person, there weren't. So I don't have a real control group, but I couldn't help but reality check this and see that this seemed to be maybe really happening. Very similarly, when a parent was exposed as a neonate, for example, there's so many reasons that came up, but tumor, a hernia, adenoid removal, in this case, surgery on foot for a congenital abnormality. They also seem to be at heightened risk for having multiplex autism in their children. And these, I'm just showing mothers here. I also saw this with the fathers, although it seemed to me maybe less multiplexed. But let's move on to the third pattern that I saw. When there was a very, very, very heavy exposure to the mother, there seemed to be an increased risk for multiplex autism. These are just 2 friends of mine who both had major abdominal surgery after a burst appendix when they were young. They both have 2 boys with very severe autism. And I also saw with some of these cases that the mothers seemed to have, when they had major histories of surgery, they often seem to have heightened degrees of infertility, although I have no way of knowing if that's true or not. Finally, repeated exposures to the father. These are 2 friends of mine who actually suffered gunshot injuries in accidents when they were younger. This father had 5 surgeries to repair the wounds, and this father had 4 surgeries after he was shot. Both of them have very, very, very severely impacted sons, and they also have completely typical children as well. And here's a father who suffered many, many injuries playing football. He was a running back for the Seattle Seahawks. His name was Kurt Warner. And he describes in the book having 6 surgeries in the very, very latter part of his brief career on his ankles and on his knees. And in his book, he wrote about having these 2 sons with severe autism. Okay, so of course, those are just anecdotes. Those don't mean anything. But again, I had to explain why I'm so interested in this idea. So when I started looking into the literature to say well, could general anesthesia possibly be damaging germ cells? Could it possibly be exerting a heritable effect? This is what I found. We all love general anesthesia. I love it. It enabled modern surgery. It's a miracle. It's a great thing, but it's extremely toxic. And I think we tend to be very cavalier about general anesthesia when in fact, it's poisonous in many, many ways. It's neurotoxic. It's genotoxic on a somatic level. It's epigenetoxic to neurons. It's reproductive toxic. It's germ cell toxic in a cellular manner. It's germ cell toxic in a molecular manner. And as I will discuss with respect to the rodent models, it also seems to be offspring toxic, although there haven't been any studies in humans yet. It's also toxic in other ways, and it's a potent greenhouse gas. So let's just go through these toxicities. So I think most of you probably know that the FDA actually issued a warning several years ago that surgery in infants younger than 3 may impact their long-term brain development and learning ability. We're also probably familiar with postoperative cognitive dysfunction in older adults. So there's really no question that it's neurotoxic. And mostly in animal models, but I think to a degree in humans, we've seen many, many, many possible mechanisms, which I will not get into right now. And this is, of course, the Environmental Mutagenesis Society. So you guys care most about genotoxicity. And here in somatic cells, there's been actually a bit of research, not a huge amount, but there's been a bit. And it is fairly consistently shown that with all of the halogenated anesthetic gases, they're all reported to increase DNA damage, both at the site of contact, for example, nose, buccal, and lung cells, and in peripheral blood. And we've seen that via a variety of different assays to measure DNA damage. The consensus, as reported in a recent paper, is that DNA damage during general anesthesia has been well documented, and it delivers a significant burden to the patients. And the extent of damage is often seen to be dose dependent. Now, I looked in our own journal, Environmental Molecular Mutagenesis. There really wasn't very much on this exposure, but there was one, for example, I believe this was from Brazil. And in this study, it found desflurane caused DNA damage in lymphocytes. Another phenomenon witnessed is that mixtures may be more genotoxic. So a trial involving nitrous oxide was shown to delay repair of damage in DNA caused by sevoflurane, and that was in peripheral blood. And there are other problems with nitrous oxide. I won't get into that gas as much. Waste anesthetic gases. So you're also probably familiar that health workers also have a risk from these drugs, just the chronic low dose exposure. And there's less genotoxicity found in this population, but they have seen in some studies strand breaks and other DNA damage by COMET assays and other assays. And with regards to neurons, it's been found to be epigenotoxic and induces epigenetic and chromatin modifications that alter neural gene expression, even absent any mutation. There have been changes seen to key transcription factors for the brain, histone modifications, DNA methylation perturbations. So general anesthesia has been seen to derail transcription of genes critical for early brain development, including cortical neuron migration, cytoskeleton, synaptogenesis, and it causes disruption of the GABAergic system. And this is, to me, as an autism advocate, is very intriguing because these are the same types of systems that are impaired in the autism brain and in the development of the autism brain. So as some leading anesthesia researchers wrote in an editorial in the British Journal of Anesthesia, we're faced with a real possibility that general anesthetics are not innocuous agents that only put children to sleep, but rather formidable modulators of chromatin remodeling and function. I won't get too deep into the reproductive toxicity here, but I think it's well established that both patients and providers are at enhanced risks for things like miscarriage and premature birth, also infertility in providers, and also to an extent cancer. We've seen an altered male-female offspring sex ratio among providers in several clinical studies with that's skewed towards females. Now, germ cells, we're finally talking about germ cells. So starting actually in 1979, researchers saw that general anesthesia, in that case halothane and nitrous oxide, was toxic to germ cells. There was another study in 1981. And most of this work has been done in sperm, although there's been surprisingly little work considering the vast extent of this exposure and the high toxicity of this exposure. But we've seen decreased sperm count, morphological abnormality, reduction in motility, apoptosis caused by these gases. Very, very importantly, we've seen damage to the spermatogonial support cells, like the Sertoli cells, with halogenated compounds generally. I don't think this work has been done specifically with regard to GA gases, but this is an incredibly important observation that could explain a lot of what we're seeing with the abnormalities in both the males and females. And along the same lines, the sperm pathologies are related to the general anesthetic's ability to disrupt normal reproductive hormones. So these cells, in part, produce the hormones that are necessary for normal germ cell development. Now, oocytes are almost always the forgotten cells, and there's been, I think, 2 studies that I could find on this. And one was in mice. It found reduction in intact oocytes, significant histological ovarian injury. And another one in rats that I mentioned before from 79 found a mix of halothane and nitrous oxide, decreased ovulation and implantation, and slowed fetal development in rats. Now onto the molecular aspect of germ cells. There has been, and this is just absolutely shocking to me, I have to say, I'm floored by this. There's been only one study that I could find of genetic damage in human sperm caused by these gases. And it found DNA damage in sperm in vitro. It was done by a group in Turkey, published in 2012. And it found 3 of the gases caused genetic damage in sperm. The effect was dose dependent, so they used various concentrations. One of them was not dose dependent. It just caused havoc no matter what. And no genotoxic effect for one of the gases, which is surprising because it's known to be genotoxic in somatic cells. No data for repeated exposures or different durations. It was a very preliminary type of study. And I could only find one study on this in animals. And again, it's that 1979 study, and it found an increase in chromosome aberrations in the spermatocytes. There have been 0 studies that I can find in the most important cells in our bodies, sorry, boys, which are the oocytes. Now, with regard to epigenetic damage in the sperm, only one study. I'm floored. I'm shocked by this. I can't believe that this is true, but there have only been one study, and it was inadvertent. The researchers weren't even looking at general anesthesia, but they were looking at sperm after bariatric surgery. This is from a group in Denmark, and they did find genome-wide changes in DNA methylation in spermatozoa, significant changes in 1,500 genes. So now let's move on to part 3, which is, okay, well, it does seem that general anesthesia can do something to germ cells. Okay, that's very nice, but does it in fact exert a heritable effect? And does that heritable effect involve brain and behavior? So far, there have been 8 rodent studies, some of which are unpublished, but I will go through them and show you what they found. Okay, we noticed these things 40 years ago. 40 years ago at NYU, a lab performed 2 studies, one in 1981, one in 1985, that reported abnormal behaviors in the offspring, right, of the exposed germ cells. So here they used halothane in utero mice, and they found impaired learning function in the progeny. This was a very short little study. They didn't elaborate very much, but they did say that it suggested that the anesthetic agent may have caused a genetic aberration in the eggs. Same thing when the sires were exposed. And then we went to sleep for about 34 years. And then more recently from the lab of Dr. Martinyuk at University of Florida, 3 studies using sevoflurane showing that neurobehavioral abnormalities were found in the male offspring of the exposed germ cells, not in the females. And they found hypermethylation both in the sperm and in the brains that were associated with the abnormalities. And Dr. Martinyuk was clear that the germ cells may be more susceptible, right, to the effects of these anesthetics than the parents or than, the somatic cells. And if you were at EMGS last year, maybe you heard him speak, and he presented unpublished data showing that hyper and hypomethylated regions were widespread in sperm from the rats that were neonatally exposed to sevoflurane. So more recently, Dr. Jevtovic Todorovic at Colorado, she exposed neonate rats to sevoflurane, and the offspring's brains exhibited epigenetic abnormalities, including reduced DNA methylation. And she didn't look actually at the germ cells, but she said that it suggested that sevoflurane causes epigenetic modifications in the early rat oocytes. Melissa Pepling at Syracuse University is using a mouse model with halothane and has been finding that male offspring of the exposed fathers are exhibiting a higher level of anxiety. And strangely, the females seem to be a little bit more social, which is a surprising finding. Finally, from the lab of Victor Corses, Dr. Wang has been doing experiments in mice using sevoflurane in utero, and the F2 male offspring, 30% of them have been found to be less sociable. Now, Dr. Corses, their lab, they're very molecular, so they're looking deep into the sperm, and they're finding many changes of chromatin accessibility in regulatory regions of ASD-causing genes. For example, changes around the ADNP gene, which is a gene common, not very commonly, but among the genes most commonly mutated in autism. And Dr. Wang observes that not only DNA mutations, but epimutations can lead to ASD phenotypes in animal models. Finally, this hypothesis checks out with many puzzling patterns seen in autism research. And I can't stress, it's incredibly rare to find an idea that actually fits all of these kind of wacky things that we're seeing with autism. It fits with a temporal increase in autism prevalence, the strong heritability of autism. And that heritability, by the way, has never been shown to be ancestral in nature. It's been shown to exist primarily in the same generation. So it's among siblings, sometimes among half-siblings. It is consistent with the neurobiology of autism, that is the transcriptional dysregulation of early brain development, the clinical heterogeneity, the parental age effects, which could relate possibly to cumulative exposure or weaker DNA repair or something else. The skewed sex ratio, we've seen males more at risk than females from this exposure. And also, I think the weird demographic patterns, we're seeing higher rates in industrialized countries and in certain regions. So the question remains, if this is happening, and I can't prove today that this is happening, but if it's happening, how significant might this impact be? Well, we're EMGS, and our co-founders warned us 50 years ago that we should be extremely vigilant about chemical exposures to our germ cells. Dr. Neal said germinal cells are what determine the health, intellectual capacity, and other prime attributes of future generations. Everything possible must be done to protect those. Dr. Crow was very concerned when he was talking about chemical risk to future generations that some compound presumed to be innocuous is in fact highly mutagenic. Today we would add epimutagenic, and that large numbers are exposed before the danger is realized. So let's do a back of the envelope, okay? If we just take 1% of inpatient surgeries and we assume that 1% result in an adverse heritable impact, and I realize like most of these surgeries are to older people, so this is very rough, that would be almost 500,000 cases per year, right, of some level of impairment caused by the germ cell exposure. So potentially we're talking about a gargantuan public health calamity that nobody has really noticed. Yet based on no data whatsoever, we are now operating under the very simplistic assumption that very toxic general anesthesia exerts no adverse germline or heritable impacts. That's the state of things today. In summary, autism rates have been increasing sharply over the past 4 decades. Causes are largely unknown. It's strongly heritable, but not strongly genetic. Autism family stories piqued my interest. General anesthesia is highly toxic and highly prevalent. It has many dimensions of toxicity. Despite warning signs back to 1981, there have been no studies on heritable impacts in humans, which is, I think, unforgivable. Rodent studies repeatedly demonstrate germline and offspring brain and neurobehavioral abnormalities. The hypothesis accords with many puzzling patterns. And this question is 40 years overdue, and I'm making the argument that it should be a top priority for genetic toxicology and for autism research. And if you're interested in this topic, we will be discussing it more on December 11th as the final symposium in the Beyond Genes conference. You can learn more at beyondgenes.org. And thank you for your kind attention.
Thank you, Jill. I appreciate that talk. While we're getting our questions in here, I wanted to ask you quickly if anybody reported on effects on mitochondrial DNA from general anesthesia.
Oh, the answer was yeah. I remember I read so many papers. I can't remember which paper said what. It's hard to keep them all in my head. Very good question. And I believe the answer is yes, but I can't remember the details.
Okay. I can dig. So we have a question from the audience. Tamara is asking if there's any evidence that the trimester or stage of gestational development is important for the extent of the effects on anesthesia. She notes the caveat, there aren't many publications. And also, are there sensitive periods within development?
Very, 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 very good question. I think, first of all, I think there must be, considering that, that the germline is entirely demethylated and remethylated very, very early in life, The in utero period is extremely sensitive for females because the primordial germ cells are stripped and rebuilt, and then they're programmed through the first year of life. In males, that reprogramming happens, almost entirely in utero. So I think it must happen. But what we're seeing, I think, from Dr. Martinyuk's lab is that there are effects across the lifespan of the germ cells, but they do seem to be more intense very early in life. So yeah, but it's very preliminary.
Well, thank you very much, Jill.